Good morning, everyone. Am I audible? Am I audible? Yeah, okay, fine. So good morning, everyone. Welcome to day 8 of bioimage analysis images to number workshop. And joining today for this session is Dr. Saravanan Palani. And it's a great pleasure to introduce Dr. Saravanan. And he did his PhD from German Cancer Research Center, Germany. And then he completed his postdoctoral training from University of Warwick, United Kingdom. And last year in 2020, Dr. Saravanan joined IISc Department of Biochemistry as an assistant professor. His lab focuses on to decipher how cytoskeleton forces are generated through actomyosin ring machinery during cell division in various cellular contexts such as yeast and mammalian model system. To answer these questions, his lab employs cutting-edge interdisciplinary approaches such as click chemistry, fluorescence microscopy, novel in vitro based on reconstitution, lipid bilayer assays, and biochemistry and molecular genetics. So welcome, Dr. Saravanan. And before starting off, I would request Mallika to tell the instruction to all the attendees. Over to you, Mallika.
Good morning, everyone. So the instructions are general that we first request all the participants to keep their audio and video off during the session. And also participants can type their question in the chat box and we will take the questions at the end. Thank you. Over to you, Dr. Saravanan.
Hi, thanks both of you. Let me share first. Is the full screen on?
Yes, sir.
Yeah, thanks. Hi everyone. Good morning. So I'm Saravanan. So I joined the Department of Biochemistry, Indian Institute of Science last year, almost a year. So our lab is majorly involved using microscope and biochemistry and synthetic biology of binding proteins. Altogether, they form a ring-like structure in the middle of the cell. And there are many things also important here. It's not just assembly and where to assemble. If it assemble at the edge of the cell, then you don't get the daughter cell, right? So in that case, there are tip occlusion pathway, where the ring has to assemble and when the ring has to assemble and when the ring is giving a license to the myosin, the motor, in order to pull the filaments so that to constrict, and that allows in the case of yeast, right, you have to have a septum synthesis should happen in the middle. So then only there will be a barricade or the closing the border between the mother and daughter cells. Otherwise, that the cells will ooze out and then they will die. So in the case of mammalian cells, it's a similar thing happens, but there is no cell wall here. It's just a membrane ingression. And then in the case of the final pinching event is done by Escort complex. And then finally, they kind of pinch it off. And then so why we use yeast as a model system, right? So why not mammalian cells? Because in yeast, as many of you know that it's a very easy to handle the system, as well as the high rate of homologous recombination and the tagging and the knockouts and also knockdown. And there are many interesting and also that available tools are there in order to understand. And most importantly, most of the genes are involved, the proteins are involved in this process are highly conserved, especially the structural proteins such as actin, myosin, tropomyosin, and these are all actin binding, especially signaling molecules such as cyclin-dependent kinases and phosphatases and so on. And so I used binding yeast as a model system during my PhD times to understand how the signaling mechanisms are involved in to decide when the cell to divide, right? Without the signaling mechanism, nothing happens inside the cell. So, and later I moved to Warwick, Professor Mohan Balasubramaniam's lab, where I used Bombay fission yeast as a model system to understand how the signal are taken up by the structural protein and how they are working at a single molecule level. So I worked on both the yeast, binding yeast as well as fission yeast. So the concept is the same, so that they make a ring. The way they make the ring are different, but at the end of the day, the ring and myosin, they are just pull, and then that's how they induce the contractility. So here, unfortunately, so far, no one has looked at actin on its own, because you cannot tag G-actin with GFP or M-cherry or any other. So far, that what we have looked actin in any of the model system, either by immunofluorescence fixed the cell or use some surrogate markers, a recent tool which came like 7 years ago, which is a LifeAct GFP and so on. So in the last 50 years, what we have accumulated the knowledge about this actomyosin contractility, it's enormous. So now we have found almost like 50 plus proteins are involving in this event. In 1970s, it's been thought it's only actin polymer and myosin and few other proteins. And now the knowledge we accumulated, it's a lot. And also the complications what we accumulated, it's also a lot, because that every protein we need to see that when they are coming into that into the actin polymer, and how they are accommodating myosin and other proteins, and how they are inducing the contractility in order to generate the tension or the force. So just to give one example, early days in my PhD, that I was working with the signaling aspect of cytokinesis, and one of the phosphatase, and which was working, it was almost 10 years ago, roughly, that I was working with the phosphatase. It's called CDC14. It's a conserved phosphatase from mammals, yeast to mammals. And this phosphatase, most of the time, kept in the nucleolus, so that you can see such a bright, as you can see at the bottom, so that it's most of the time it's in the nucleolus, and then during the late anaphase it comes out of the nucleus. There's no problem. People have seen this enough until then. So there was an evidence that this phosphatase can also play a role, not only exit for mitosis by down-regulating cyclin-dependent kinase, they can also induce cytokinesis, but there was no evidence at that time. So that was the time Delta Vision was making a major impact, especially with the budding yeast and especially with the yeast community. So the same strain I was using, CDC14 fused with C-terminal GFP, I was looking at it. At that time, in my own lab, that we had a mercury lamp, then we are not able to see what you see the localization here at the division site. We are not able to capture that. So everyone thought that maybe it doesn't play any role in cytokinesis. So when I was using a Delta Vision imaging system, where the same fluorophore, nothing is done. So then you can nicely see that it localizes roughly 4 to 5 minutes, not more than that. But it's very difficult to capture in any other system. That's why I'm saying that. So when you are talking about microscope, especially a fluorescent microscope, it is not just a system. It is not just a fluoroscope. It's a combination of everything. And this is just to give an example that why people have missed, because this phosphatase was identified by Lee Artwell, is one of the Nobel laureates. But until then, everybody was thinking that this phosphatase play a very major role in exiting mitosis, but not for cytokinesis, because there was a limitation that we are not able to look at them at the bud neck, where the cell division happens. At the moment we have seen that it localizes to the division site, then we look for the substrates, and then so on and so forth. So, so as I said, so people are thinking that this phosphatase, it's under the mitotic exit network, is one of the conserved network, and what known until 2010 or 2011, that this phosphatase play a role in mitotic exit, not in cytokinesis, because of the limitation of the microscope as well as the tag and how to look at them. So then I figured it out that it localizes to the division site, and then followed by several biochemical yeast-to hybrid and so many assays. So make the story short, that it's not only the phosphatase playing a role in mitotic exit, it's also playing in cytokinesis by characterizing a couple of substrates, because when this phosphatase localizes to the bud neck, and that's the time couple of substrates are dephosphorylated, and that is a very key event for cells to ingress the ring, that means that constrict the ring, as well as important for the cell separation of it. So then I moved on to my postdoc, where I've used a microscope much more than my PhD time, where I was looking at a single molecule level, where the complication starts. You know, like when you're looking at the live cell, it's fine, but when you're going at a single molecule level, also the fluorescent microscope, the turf-based microscopes, these all play a very major role to choose the right target as well as the tags. So here, that as I said, that the structural unit, that you have a polymer, it's like a track, and myosin is like a train, and then you need other tools such as tropomyosin, which decides when the train can sit on the track or when not. You know, so that's the kind of a thing that what I was looking at more closely during my postdoctoral time. So as I said earlier, that I've used Pombe as my model system. The reason is why Pombe, because Pombe, the actin-myosin ring, it's almost like a 4 micron. In case of budding yeast, it's 0.9 micron, and it's a very good imaging system. You know, like naturally you can see very beautiful ring. As you now you can see the previous movies compared to budding yeast, that the Pombe is as got a bar, so on. So the next thing, from my postdoctoral works, I'm moving from the confocal microscope, confocal spinning disk, to the turf microscope, because when you do an experiment or a project that, you know, especially when it comes to microscope, especially fluorescent microscope, nowadays the demand is more, right? So you have to always, you know, excel yourself, or you have to keep on learning more and more and more. And then this is what happens to myself as well. So one of the project I was involved working with the non-muscle myosin motor. So we have shown very clearly the defective, as you can see on the top, where you have the wild type cells, it nicely forms the ring and then constricts. And one of the myosin defective, that means the motor defective mutants, they fail in assemble the ring as well as the constrict, and then they stayed for almost 3 hours. Normally this whole process happens within 40 minutes, but it took almost 3 hours. But still, the defective myosin mutant not able to form a ring or to constrict, it stayed and then just disappeared, or it's kind of the bundles are just falling out. So we have to show that in vitro. So how will we do it? So one of the experiment what we did was that we purified this myosin, that means the endogenous myosin, along with the light chains, because myosin it's a heavy chain, works with the 2 light chains, which is the regulatory as well as the essential light chain. So we purified this myosin from the fission yeast as the original host source, and then tether them on the polylysin or nitrocellulose coated thing, and we put the myosin, and then we throw the actin on the solution. So as you can see on the top in the wild type, so as you can also see the tracking, the actins has been captured by the myosin, and then it's kind of keep moving, and then that's what you can see that the actin filaments are kind of swimming through, because it's a wild type myosin can able to attach to the actin and then move the actin filaments. But in the case of defective, as you can see, though we have the myosin, we have shown that the myosin has been purified and attached to the slide, but what we are not able to see here, that is very nicely correlated with our in vivo data, that is not able to, even first of all, it's not able to capture the actin, because if it would have captured the actin, you would have seen the actin gets captured and we would have imaged them. But it's not able to capture. What you can see is just a kind of a jugglery. That means the actin is kind of just floating on the solution up and down, because if the myosin is defective, not only it's a motor defective, it's also having an attachment defective, right? So that's one thing. And other important part during my post time, what I've done is that, so what we know about this actin polymer, and then the polymer is being protected, because when actin, in the G actin, it's a monomeric actin, becomes the F actin by FORMIN, right? So this particular F actin needs to be protected from severing factors. Otherwise, we don't have actin filaments, we don't have cell shape, we don't have anything. So that has to be protected. So there is a protein called tropomyosin, and which is known in the smooth muscle cells, that this actin polymer has been wrapped over by this protein called tropomyosin, and which masks the myosin binding site. When there is a stress and the calcium level goes high, and what happens is that myosin masked site has been released, and then it can be exposed. Then the myosin comes, binds, and moves the filament. That's where we have this stress and relaxed in the muscles. But this is known in smooth muscle cells for a very, very long time, almost 30, 40 years. But what is not known until 2018, that how it works in non-muscle cells, right? This is smooth muscle cells. Okay, you have a calcium, and then you have this tropomyosin, troponin complex, and all that. But in the non-muscle cells also, do you have actin, you have a tropomyosin, you have a myosin. But why it is not regulated in that way, in that manner? So this particular regulation is completely unknown, how it binds and how it not binds. So what we found was that one phosphorylation site, which is present in the tropomyosin, and that kind of regulates when the tropomyosin to bind stronger, and when the tropomyosin has to leave the actin polymer. So in order, this will be exposed to the actin severing proteins or other proteins in order to come and act on them. So in this case also, that we have to use a turf-based microscope. So here the problem comes, right? So as I was telling that in the beginning, that many of the actomyosin or the cytoskeletal protein, tubulin, actin, tropomyosin, alpha-actinin, there are so many proteins you can name them, and all of them are kind of, you cannot tag them like in a conventional way of tagging, either the GFP in the C terminus or M neon green or M cherry N terminus. Nothing works, because these guys are all forming a head to tail dimers, right? If you tag the N or the C, you always have a problem. Okay, so you have to use a different way of tagging them. That means, marking them with the fluorophore in order to do what kind of experiments you want to do. For example, turf microscope and so on. So in this case, that I wanted to visualize tropomyosin on actin. Both of them are untouchables. You cannot tag them. So then we use a cysteine-based maleimide labeling, which is many of the single molecule people at the minute are using, and it's very easy to use it because there are commercial dyes. You have to introduce a cysteine which is far away from an endogenous, so that you should not interfere with that endogenous disulfide bond. So you can put a linker and have a cysteine, and then you can label them. and then in the case of visualizing. So in that case, I have to do for both for tropomyosin as well as for actin. So what we have done here is, you can see here that the tropomyosin is in cyan and actin is in magenta. So we have shown that I can play this movie again. So we have shown that this phosphorylation, it's a key event when the tropomyosin to bind and when not to bind. And we found a kinase which is responsible to put that phosphorylation to remove the tropomyosin from the actin. And then we also used the kinase dead of it to show as a control that this is happening by this one specific phosphorylation site, by that one specific kinase. So what we did, so we labeled both actin as well as tropomyosin using the cysteine maleimide labeling, click chemistry based. And then we put it on the turf microscope. As you can see, if you add the wild type, immediately the tropomyosin is taken off when you add the wild type kinase. But if you add the kinase dead, nothing happens because there is no phosphorylation is happening because you added the kinase dead. And one more control we added, if you mutate that serine to the alanine, where it cannot make the phosphorylation, even in the presence of wild type, nothing is happening. So that clearly tells that this phosphorylation is the key event in order to remove the tropomyosin out of the actin filament. And this nicely we can visualize using the turf-based microscope by labeling. Here where the limitation, as I was telling, that you cannot label them in a normal way. So you have to label it in a different way, so that you have to use a cysteine and maleimide based labeling. So the other thing that, as I said, this actin filaments needs to be protected. That's the reason tropomyosin binds and protects. If the tropomyosin is not there, so then it will be susceptible to any actin severing proteins in order to come and then sever the filaments, and then the cells leads to lethality, right? So that's where like you have a polymer made, and the polymer is protected until some time, so that cell needs to achieve certain jobs, because most of the motors have to walk through, and also the cells to attain certain shapes. For all that filament maintenance is very important. That's why the turnover in the case of actin polymers is very, very important. Otherwise, they will have a very severe effect on cell growth as well as the cell morphology. So to test this further, again, the similar thing. So we had a drug called the sphenolyde, which severs the actin filaments. So if you don't have the actin filaments protected by the tropomyosin, the actin binding protein, so the moment you add the drug, you can see that actin filaments severed off. But in the case of the wild type, it is not. But in the case of the E mutant, where it binds weaker with the phosphorylation mimic mutant, where you can see that the filament gets severed off because the binding efficiency between the phosphomutant and the actin is very less. So that's why the drug can easily act on it, and then it kind of severs the filaments. But in the case of wild type, the drug cannot, because it forms a very tight complex. So, so this is all the part that what I was talking about in vivo and kind of in vitro. So my research mostly revolves around that actomyosin ring. So how this actomyosin ring is useful in the case of cell division at different organisms at different stages of the cell cycle and so on. So one question which is always there for many people, that can you make a synthetic cell? Can you make a can you make a synthetic cell and then it can divide on its own? For that we need to know many things, right? First of all, that we need to know how many proteins are there in order to make a ring-like structure. So what we know so far, that actin is a straight filament. What you read so far in the textbook is actin is a straight filament, right? Actin, so far, I don't think any book has shown or any articles has been shown that actin can also bend and form a ring. If actin is not bending, how do you achieve the ring-like structure, or how do cells achieve different shapes if actin is a straight filament, right? So so very, very first experiment, that this is what we have seen over the last years. The very first experiment, or the sea urchin, was used to understand cytokinesis, where they injected the myosin antibody and then localizes in early 90s and 2000, that they showed that sea urchin has a model system where myosin localizes to the middle, the cleavage furrow, and that's where they decided that, okay, this particular protein, it's a key important protein which is important for that cell to divide. And then later that when the tools are coming up and a lot of new advancement in the microscope, and where we can see decipher the or eliminate a lot of complexities and then put a lot of knowledge into it, and then we understand much more about the cytokinesis or how cell division works, even in a complex organism such as the sea urchin. So during my postdoctoral time, that we achieved one thing, is that we isolated this ring apparatus from the cell. Okay, we not studied only in the within the cell. We isolated, and then when we added the ATP, and then the ring nicely constricts. And whatever we isolated, the ring-like structure from cell is an active complex, right? So when you add ATP, and then it's nicely constricts. Okay, so this particular ring isolation system and its ATP dependent constriction works in fission yeast cells. So the similar thing what I did was using a budding yeast system, because budding yeast also form a ring-like structure, and it's also in the division apparatus. Arrested the cells, and then we kind of perturb. There is a different protocol in order to perturb the cell wall and poke the membrane. And after a small centrifugation, short spin, what you can get is that the ring just came out of the cell, and if you centrifuge, all the cell debris goes to the pellet, and then the ring just floats on the supernatant. If you take the supernatant after the centrifugation and put it on a cover slip and visualize in a microscope, you can see a ring-like structure. And this is what we are going to use as a template to study more. So all this knowledge gave us one thing. So, what is the key proteins are involved to make such an elegant ring-like structure? How many proteins? Do you need 20 proteins, 50 proteins, 100 proteins? If you look at any cytokinesis or actomyosin ring reviews in the last couple of years, there will be always a list, or in the supplementary table in the reviews, you can see that almost like 100 proteins or 120. The list is always going longer and longer, but we don't know till date that how many proteins do you really need to make such a ring. Okay. So in that case, going back, as I said, for a muscle, you need 20 proteins in order to achieve this contraction business. But in the case of budding yeast or a fission yeast, what you need 100 to 150 proteins. Can you narrow down to a certain number of proteins? Can be able to reconstitute a synthetic division apparatus on a test tube or on a cover slip. If we manage to do that, that will be one of the biggest thing that what we can say that can you make a synthetic cell now which can divide on its own and then propagate a daughter cell, right? So in this aspect, we were looking at it. So we've taken a different approach and not imaging on a cover slip based turf bias microscope, so that we were using a supported bilayer system. So supported bilayer system also used by many biophysicists in order to study that myosin dependent constrictions and so on, pulling the actin filaments and study the membranes and so on. So we have used a supported lipid bilayer system in order to tether the protein of interest what we want, and then we can add on top of the other proteins and see that are they making some kind of a structure or not. So as I said earlier, that what we know so far, if you take any actin binding protein, for example, here I mentioned two, azirin actin binding domain or a utrophin actin binding domain. If you incubate the actin, F-actin, in the presence of any of this protein, it always makes a very nice bundles as a straight filament. You never see them, or you are never shown in any of the books that actin could bend and then kind of a curve the actin filaments in the presence of any other protein. So we made a discovery. It was 3 years of work. It was just published 2 weeks ago. That there is a conserved protein called IQ-GAP1. It's present in yeast from mammals. Then it has got a similar way of domain architecture and so on. And it has got an N-terminal CHD domain. It's a calponinin homology domain, and followed by the USR, this unstructured region. So as I'm sure many of you know that, as I said also earlier, that you cannot tag actin. So people have used LifeAct as a surrogate marker in order to mark actin structures, either filaments or a kind of a patches and so on. So before LifeAct generation, people have used this particular part, the CHD and the unstructured region, as a marker. They normally take this particular domain, almost like 200, 250 amino acids, and fuse with GFP and express in mammalian cells, express in yeast cells, and to mark actin filaments. Once the LifeAct has come, then people started using less of this, and then they were using LifeAct. But what they've missed is that, so whatever people have used so far using this as an actin binding tool or actin marking tool, so even myself use this always on a cover slip or expressing them in yeast cells and mammalian cells and marking it. So we just, along with one of my postdoc, Darius, who's a biophysicist, who's an expert on this, putting this lipid bilayer thing. So we decided that to try something new, why don't you take this particular piece? Because no one has studied, so the entire domain organization of IQ gap, because this protein is known for last 40 years and people have studied left, right, and center. They know that each domain does what and all that stuff, either in yeast or in mammalian cells. And also that this IQ gap, because it plays a very major role in many cancer cell shape regulation, right? And means that exclusively studied protein, but one part people have missed to study is this unstructured region. So when we purified only this 1 to 189, that means that 190 amino acid stretch from a yeast part, and then we put with the nickel, so that the lipid bilayer has got a nickel NT, how you normally purify the nickel lipids, and then we purified the protein with the 6S tag. So this can be captured by the supported bilayer, and then be imaged by throwing the actin, because we know that it's an actin binding protein. So we didn't expect, we thought it will bind and then it makes some kind of filaments or bundles and so on. So surprisingly, what we observed was that it is making a ring-like structure, just two proteins. Okay, so supported bilayer, and then we put this particular domain, that 190 amino acid, and then we throw a pre-made actin filament, and then the actin filaments was coming and attaching to this protein, and then this particular protein making the curl, and then makes it like a ring-like structure. So the next thing is that, okay, is it an artifact? Because you made a pre-made filaments, and then you are kind of throwing it, and then maybe it's something is, the buffer or something is inducing this particular structure. So then to avoid such questions, what we did was that, as I said, actin has been bind to propylene, and it's taken to the formin, and formin makes the actin filaments, right? So then what we did was that we tethered the formin this time to the lipid bilayer, and then we throw a G-actin in the presence of a curly domain. So we named it curly because it curls the actin filament, so that we named it curly, the CHT and the unstructured domain part. So when you tether and then you put the G-actin, as you can see in the beginning of the movie, there is no filament, right? So when you can see, when the actin filament is polymerized by the formin, in the presence of a curly domain, it gets curled, and then you can see a nice ring-like structure. And this is not the case when we add any other actin binding protein. We have performed the similar experiments with ezrin, utrophin, Lifact, and different actin binding proteins. The similar set of experiments in the presence of formin, it never bends and never curled it. So once we made this, we were also surprised to see such a elegant way of making a ring-like structure. This is the first evidence, as I said. So far, no one has shown that actin could bend and then makes like a curl-like structure. And then we got a confidence that, okay, this is the way that one can make a synthetic actin ring, like completely a reconstitution. There is no other. We know that the picomole, the nanomole of the protein amount we added into it, and then we know how much the proteins are there, and we can make such an elegant ring-like structure. So the next question obviously come in is that, can you make them constrict? Making a making a ring-like structure is easy now, looks like. So can you constrict? Until or unless if you cannot constrict, you cannot call them as an actomyosin ring constriction. So what we did was that when we made such a ring-like structure in the supported bilayer, the next thing is that we throw a myosin, the purified myosin, into it. So now we made it a 3-protein complex. So we had a curly domain, an actin that could bend and make a ring-like structure, and then we added a myosin, purified myosin, to it. As you can see, the ring kind of constricting. So in the beginning of the movie, I'm sure that you would have seen that there are many rings, and now the end of the movie, now the movie is playing again. As you can see, there's a very nice ring-like structures, and then the myosin is added, as you can see here, the end, the bottom. Then you can see many rings were constricting, and then this is one of them. As you can see, it's nicely constricted. So now we come to a conclusion. Okay, now we can surely say that we can bend actin filaments, and that can make a ring-like structure. And this can be enhanced, as some of the data I'm not showing, that it can be enhanced or you can be dismantled by adding the friends of actin filament and the enemies of actin filament. If you add the enemies of actin filament, what I mean is that they won't allow them to bend, they kind of make them branches. So even in the presence of the curly domain, it won't bend, it kind of turns off and then it makes branches, it never stops. And if you add a friend, such as the tropomyosin or alpha-actinin, such proteins enable them to bend much more elegant way, so that they can make a ring size almost like how cells make it, almost like 1 micron or kind of 2 micron and so on. Okay, so the next question comes. All these experiments were done, purified protein from an equal comes from a fission yeast. Okay, IQ gap. And the next question comes: is this conserved? As I said, IQ gap is conserved from yeast to mammal. Is it conserved? And this particular unstructured region of IQ gap, can it bend from different organisms? So what we did, so I purified this particular IQ gap. So in Pombe it's called ring 2. In budding yeast it's called IQG1. In human it's called IQ gap. So then I purified this particular domain, which is the CHT domain, the Kalpanin homology domain, followed by the unstructured region. So the unstructured region is you have different